I'm going to start by giving you just a little bit of background on our Pancreas Center at OHSU, because one thing we're trying to do with these meetings is build collaborations, and since we have a new center, I want to tell you just a little bit about it, and then I'll move into a small story that has come out of that, out of some of our efforts actually in our Pancreas Center. So our Pancreas Center has three clinical and research focus areas: early detection, advanced therapy, and quality of life. And we have a large pancreatic tissue registry that was founded by Brett Shepard in 2007. And what we've done recently with some, as we're building our center, is really try to take a deep dive into analyzing samples from our patients, and we have worked very hard at putting together the infrastructure in which we can acquire serial blood draws as well as tissue or biopsy samples from our patients during their clinical course. And from those patient samples, we're trying to take a really deep dive in analyzing them. And so through a team effort that involves Lisa Cousins and Joe Gray and many other investigators at OHSU, we've been really trying to do deep analysis on our patient samples. So Lisa and Joe both have two different platforms for doing multi-plex IHC or IF to look at either immune cell components or other stromal cell components in the tumors and the tumor cells themselves. We're doing DNA and RNA. Joe's group is doing some electron microscopy work to look at tumor ultrastructure, which is proving to be very interesting in terms of how these cells talk to each other. We also have blood assays. We have a new collaboration with a company called Immunovia that has developed an ImmuArray platform. Their discovery chip has 500 single-chain antibodies oriented on it that detect cytokines and blood-based proteins that you can use to make protein signatures that they now have what they think might be an early detection marker for pancreatic cancer. And then one thing that I'm particularly excited about is we've been preserving live cells from all of our patient samples. So we started doing what everybody's been doing with the PDXs, and actually we've been making organoids for many years now. One of our faculty trained with Hans Clevers early on, and so he's been making PDAC organoids for the last 5 or 6 years. But then more recently, we've moved to a new culturing technique that we call the J2 co-culture technique that comes out of technology developed by Dick Schlegel at Georgetown. And I'll tell you a little bit more about this because we're getting 100% efficacy in the ability to grow out and preserve live cells from our patients. So we're excited about that. So from that large amount of data that we're generating, we need to be able to relate it to the clinical information over the course of the patient's treatment, since we are collecting serial blood and tissue samples. And so we're using a lab key database that was developed out of the Hutch, in which we can both include all of the clinical data as well as all the research data, including image data as well as pipelines for the omics. And the nice thing about this database is you can scrub PHI from it, and then we could share it. And so one of the things with this consortium, we want both SOPs for standard practice in terms of collecting samples so we can share them. And then also it's really going to be critical to be able to share the data, including deep clinical data, across the consortium or collaborators all over the world. And so we're very interested in making a robust database in that sense. So just a snapshot, we've developed 30 PDXs, and we've actually kind of stopped doing this now. We just preserve the cells so we could make them if we want to, but the ones we have a diverse histologies and diverse in terms of onset or recurrence after resection, and so we have a spectrum of different PDXs. This is our culturing system that I told you about. We had been doing the organoids for quite a while. One of the things we find with the organoids is we get about a 67% take rate, which doesn't quite serve our needs because we really want to preserve cells from all of our 100% of our patients. And that's why we moved to this J2 co-culture system, which now we've validated. We have many several more, but we've validated through STR that it is patient, that we're deriving the cells from the patients, and through mutational analysis that they're KRAS mutant, or that they show the same mutational spectrum as the patient sample. So we know these are validated cell lines that we've grown out, and this has actually just been an effort in the last 6 months. So we do about 2 of these a week, where we are collecting samples from the patients and analyzing them, and now being able to grow live cells, and we can do this from serial biopsies from the patients is our hope. So we can see how the tumors evolve and develop resistance, and then we can use these once you establish the cells on these co-cultures, and also with the organoids, we've developed the technology so that we can now then plate them they last for 6 or 7 passages on just plain plastic. So you can move them from this, where they're renewable, and you can freeze back early passage, and you can keep bringing them out and growing them, to move them to just straight plastic for 384 well siRNA or drug screens. So I just want to point out that Nick from my lab has a poster on work that he's been doing with these primary early passage patient cultures. So I encourage you to go take a look at that. Another thing that we're doing is we're doing these, as I mentioned earlier, these large field IHC or immunofluorescence assays, which are multi-plex, to look at different cell phenotypes and heterogeneity. We're particularly interested in heterogeneity across the tumor, both in terms of the stromal compartment as well as the tumor cells themselves. And so we're doing phenotypic analysis of different aspects of the tumor cells, including metabolism, cell lineage, but then also the stromal cells in terms of ECM and immune components. And another nice thing that we've worked out is on one section we do this large field image with multiplexed IF, and then on an adjacent section we co-extract RNA and DNA, and we can do RNA-seq and whole exome sequencing from that FFPE section. And so for the RNA-seq, I mean, that's not so hard for whole exome sequencing, but RNA-seq, I was very skeptical to start with, but we're using an exome capture method that can take the RNA from the FFPE and actually give you very high quality RNA-seq data. And so we're now starting to do that. And the nice thing about that is you can macro or micro dissect regions of the tumor that has interesting phenotypes. And then this also, Jason Link has a poster on these efforts, and so please take a look at that. So one of the things we're looking at with our multiplex IF platform is heterogeneity, as I said, in the cells. We're very interested in the idea that cells can have lineage or differentiation state heterogeneity. This work really stemmed out of some of our breast cancer work, where we found this type of lineage differentiation state heterogeneity really impacted response to therapy. And so we're very interested in this. So just a little primer on the differentiation of the pancreas, and really the main thing to know is you've got a common stem cell, and then you have a bipotent progenitor coming out of that, which can give rise to the exocrine ductal compartment as well as the endocrine islet cells. And so we were interested in these different lineages and started using lineage markers to see if there was heterogeneity within the epithelial compartment for different markers for these different lineages. And this brought us to what we thought was very interesting, which is that when we looked at, used a pan-cytokeratin to look at epithelial ductal markers, ductal cells, and then also used a synaptophysin to look at as a neuroendocrine marker to look at the neuroendocrine lineage. We found that in quite a number of our patient samples, and depending upon how we do our cutoff, it could be anywhere from 20 to 40% of patients with actual PDA, pancreatic ductal adenocarcinoma, they have co-stained cells within their ductal lesions that co-stain for pan-cytokeratin and synaptophysin, and that look to be tumor cells. And so we're really interested in this, particularly with some of the literature that's come out in terms of non-small cell lung cancer being able to transdifferentiate into small cell lung cancer when it becomes resistant to the therapy. and then also in prostate cancer, again, a ductal adenocarcinoma transdifferentiating to a small cell neuroendocrine tumor, very aggressive, kills the patient rapidly, and this is when it becomes resistant to the therapy. So we were interested in this idea that maybe this was indicative of a underlying lineage plasticity, which could be a poor outcome marker. And so this work is all being presented, and this is where I'm going to spend the rest of my talk talking about this story, and it's being presented in a poster also this afternoon at lunchtime. So I had a surgery fellow in the lab who did this. He looked through a lot of our surgery patient samples and tried to find a matched cohort, and he ended up here in this study with 16 matched patients where they all had an R0 resection. They all received standard of care gemcitabine at the same timing after that resection, and they were well matched for gender, age, stage, and grade. And from these patient samples, he then scored their tumors based on the percent of co-stained cells where he was co-staining with pan-cytokeratin and synaptophysin, and then looked to the days of recurrence post-surgery. And what he found was really a pretty striking difference. If you had higher levels of co-staining, you had higher levels of co-staining, you had a much shorter time to recurrence. And in fact, these patients are often recurring while they're on gemcitabine, standard of care, compared to patients that had a lower level of this co-staining. And then in terms of disease-free survival, again, a very significant difference depending upon the level of co-stained cells. So we also, I was talking to people at a couple meetings about this, and Andy Rim had said, oh, he's been looking at this too, and he was looking at it in circulating tumor cells, where he was finding that some of these circulating tumor cells that he was isolating were actually co-stained for pan-cytokeratin and synaptophysin, so he sent us some of his data on that. And then we wanted to take a kind of an approach where we weren't just looking at one marker, and we said, well, could we get some kind of gene signature for neuroendocrine differentiation state? And for that, we didn't have one in the pancreas that I could find, and so I turned to our prostate group, and there was actually just recently a prostate gene signature that was published. It's about it's based on about 800 genes that are that can differentiate between prostate ductal adenocarcinoma and prostate neuroendocrine tumors. And so we used that gene signature, and we used gene set enrichment analysis. And so what this signature of is these genes that are up in the neuroendocrine versus the ductal. So these genes that are up, and then we ran them through Andrew Biancon's different subtypes, and we found that only the squamous subtype was this neuroendocrine gene signature highly enriched, and not in any of the other cell types, subtypes, even if you compare them individually or lump them all together. And similarly, the genes that are down in the neuroendocrine tumors are up in the ductal cancer those are enriched in the other subtypes, not in the squamous. And of course, we initially thought, well, they would be enriched. Well, so let me just also step back and say it's the squamous subtype that are the poor outcome in those subtypings. We initially thought, well, maybe it would be enriched in ADEX because that stands for aberrant aberrant differentiation exocrine endocrine. And so, but we actually found that there was actually de-enrichment. It was enriched in the other because it's really enriched in this squamous subtype. And then again, at some meetings, I was talking to Howard Crawford about this, and he said, oh, well, and he's already published or talked about this and published a bit on this. He's very interested in some of these cells, as is Stephen Leach. And so he had done lineage tracing where he's using KCY mice. So these they are driving KRAS, and then they have GFP in the Rosa locus with a lock-stop lock, and they're driving up with a P48 CRE-ER. In this case, they're they're treating the mice with tamoxifen at 8 weeks of age. So they're basically turning on actually it's YFP, sorry. They're turning on YFP in the acinar cells in the adult mouse, and they're also turning on KRAS. And then they watch these lesions progress, and they see that they get these synaptophysin positive cells either embedded in the duct, or they can have these single cells that have kind of left the ductal lesions and are sitting here in the stroma adjacent to the ductal lesions. And so this would suggest, based on this lineage tracing, that these cells can derive from acinar cells that have undergone an acinar to ductal metaplasia, and then perhaps a ductal to neuroendocrine transdifferentiation. And then because I work on MYC, and sometimes I'm a bit MYC myopic, we know that MYC is involved in cell plasticity, and so we said, well, what's the role of MYC? Is there a role for MYC in this phenotype? And so just as by way of introduction, we did some simple experiments. You knock down MYC, and you can decrease synaptophysin expression in some standard PDA cell lines. So at that point, again, this is a very collaborative project. I was talking to Owen Sampson, who's at the Beats, and I said, well, we're interested in this lineage plasticity and trying to understand it, and is there a role for MYC? And he started saying, well, when he has the KPC mice, where he has one copy of the MYC locus knocked out, and he's shown that those mice have a longer survival. And he said they look more differentiated. So he thought, well, maybe they'll have less of these cells, and they do actually have less of these dual positive Cytokeratin-Synaptophysin cells in the tumors or in the lesions from the MYC-floxed mice. And also, the other thing we got is we got a ChIP-seq data from Martin Eilers, where we're looking at MYC bound to promoters, and we asked if we rank genes based on upregulated in the neuroendocrine tumors. This is, again, prostate versus upregulated in the adenocarcinoma. The MYC sitting on the promoters was highly enriched in genes that are driving a neuroendocrine differentiation state. And then as he was initially published, just the curves, and then he sent us some of the H&E, which shows you that if you knock out a copy of MYC, your progression of disease is severely impaired, and you have a longer survival. So based on this, and then also other work that we were doing, and we actually started doing this quite a while ago, we asked, could we make a model of pancreatic cancer based on our knowledge of MYC. And so my lab has been studying MYC and its post-translational regulation, and we know that kinase cascades downstream of KRAS can phosphorylate MYC on a site that both stabilizes MYC as well as increases its transcriptional activity, its DNA binding and transcriptional activity. And we find that we have antibodies to this, and that this phosphorylated form of MYC is upregulated actually during ADM, and then it is upregulated in early PANNs and in PDA. And so from this, we decided to make a mouse model. We started with PDX-CRE, we moved to P48-CRE, and now we're moving to P48-CRE-ER. And then we have the lock-stop-lock KRAS. And what we have is we've knocked MYC into the ROSA locus downstream of a lock-stop-locks. And we made these mice quite a while ago. We've used them for mammary cancer models. We know the ROSA promoter is actually quite weak, and so we get low levels of MYC expression. So if we look at the survival of these mice, we see that MYC alone in this tissue, as well as any other tissue we've tried, if we use wild-type MYC in the ROSA locus, it's expressed at such low levels it's not able to drive tumors at all. So this is a sub-oncogenic dose of MYC. But this deregulated low level of MYC can dramatically accelerate tumors in the KC mice. So here's the KC mice. This is KRAS only. 1 copy of MYC, 2 copies of MYC, and we progress through to pancreatic cancer in this model. And if we look in this model, we actually do see a lot of these dual lineage cells where we have pan-cytokeratin and synaptophysin on. And you can see compared to the KC only that even if you match for pan and grade, we get an increase in the numbers of these double positive cells when we're driving them with this low deregulated level of MYC and KRAS. Coming to, my timer went off. I think the battery died. That means I can talk for a long time. No, I am almost done. So these mice, like the KPC mice, the beautiful KPC model, this model is also gemcitabine resistant on a very small number of mice, so we need to do more of this. But this brings me to the question of what is the relevance of these cells. So I started by showing you that patients who have a high percentage of these co-stain cells have a shorter time to recurrence. They are getting standard of care gemcitabine. So is there any kind of does this plasticity in some way make the cells more resistant to therapies, as starting just looking at the standard of care gemcitabine? And so we just looked at the cell lines that are available, and we see that there's very high, actually high levels of synaptophysin and other neuroendocrine genes in the more poorly differentiated squamous or quasi-mesenchymal subtype of PDAC using these established cell lines. And the cell lines that have the most expression of these neuroendocrine genes are the most resistant to gemcitabine. And interestingly, we found that if we then look at the cells themselves that are resistant that remain with treatment, these cells have upregulated some of these neuroendocrine markers. Here's just a couple of them right here. So again, could this be a resistant state? We're interested in that question. Here's PANC1s. Again, they upregulate these cells. If we knock down MYC, we lower the degree to which this synaptophysin is turned on. And then we've been approaching trying to find ways of targeting MYC. And the BET inhibitors, we've been using those too. But another area of focus in my lab is the idea of using a PP2A reactivation therapy to try to dephosphorylate MYC and turn it off in that manner. And that has led to a collaboration with Gautam Narla, Case Western, and Michael Olemeyer at Mount Sinai. Michael's the chemist who developed this small molecule. So we started this work actually many years ago using a peptide mimetic that would inhibit an inhibitor of PP2A, relieving PP2A, which is a major tumor suppressor in the cell, responsible for turning off multiple oncogenic pathways, including MYC. And we used that peptide as a therapeutic for a while and published some papers on that. But now we were excited to find that this chemist had developed a small molecule. They call it the small molecule activators of PP2A. This actually physically binds to the structural subunit of PP2A and causes a conformational change in the catalytic subunit that prevents it from being turned off in tumors. And so we're actually not increasing the level of this tumor suppressor. All we're doing is preventing it from being turned off. And so we, so far in all of our mouse treatments, have not seen any toxicity with this compound, which we're excited about. So if we treat with this compound, we can decrease MYC levels, we can decrease MYC phosphorylation, and we can actually reverse the upregulation of these neuroendocrine genes if we treat with this in combination with gemcitabine, and we get a nice synergy in terms of cell kill. And then we have been treating the gold standard mouse model as well as our own mouse model. It works there too, but the gold standard mouse model, the KPC, with this drug, this small molecule, and so far very small numbers, but we're excited about it because we're getting a really significant increase in lifespan, in survival in this aggressive mouse model. And again, there's a poster that will be presented at lunch on these small molecule PP2A activators. And this is my lab, and I want to thank everybody. I've mentioned, so people here who have posters, Nick, Jason, Brittany, and Megan, unfortunately, is I need to get a picture. She wasn't in the lab yet when we took this picture. And then I also want to end with saying that we're really excited about trying to make a robust group of collaborators up and down the West Coast. And if you're not already on the email list, which you probably are, but if you happen to not be, you can write to Dov Keith or me, and we can get you on the list. And thank you very much.